Buckle up, everybody. Today we're taking a deep dive into a treasure trove of Nobel lectures spanning over a century of scientific breakthroughs.
It's a fascinating mix of physics, chemistry, biology, and even a touch of history of science,
Yeah, it's going to be a wild ride through scientific history.
and a chance to see how these different fields connect and influence each other.
Absolutely. Have you heard of Marconi?
Oh, of course, the pioneer of wireless telegraphy.
Right. Back in the early 1900s, he was convinced that the Earth's curvature wouldn't stop radio waves.
Imagine that, thinking radio waves could just zip around the planet.
I know, right? And we'll delve into how his early experiments led to that transatlantic transmission.
A pivotal moment that changed communication forever.
Totally. And then we'll jump into the world of genetic engineering.
Ah, yes, Daniel Nathans, and the revolutionary discovery of restriction enzymes.
These molecular scissors, they're like tiny tools that can cut and paste DNA.
They opened up a whole new world of possibilities for studying and manipulating genes.
Gene editing, gene therapy, genetically modified organisms, all thanks to these amazing enzymes.
Truly groundbreaking stuff.
Okay, then we'll tackle a debate that raged for decades in neuroscience: the structure of the nervous system.
Golgi, the scientist who developed the technique to visualize neurons, actually argued against the idea of the neuron as an independent unit.
It's kind of ironic, right? He made it possible to see neurons, but he didn't fully embrace the concept of the neuron doctrine.
It shows how science is a process, a journey of exploration and debate, with interpretations evolving over time.
And sometimes the pioneers themselves are reluctant to embrace the full implications of their discoveries.
That's part of what makes the history of science so fascinating.
Totally agree. And we'll also explore the strong force, one of the fundamental forces of nature, which governs the interactions of quarks.
Quarks, those tiny particles that make up protons and neutrons.
Exactly. David Gross's work on asymptotic freedom helped unravel the mysteries of this force.
Asymptotic freedom, that sounds pretty complex.
Well, it essentially means that the strong force gets weaker at short distances, which allows quarks to move more freely within a nucleus.
Oh, I see. That explains a lot about how those particles behave.
Right. It's like a strange dance of attraction and repulsion at the subatomic level.
And it has profound implications for understanding the universe, from the formation of stars to the very nature of matter.
And lastly, we'll delve into the origins of life itself.
Howard Temin and his protovirus hypothesis.
He proposed that retroviruses, those sneaky viruses that insert their RNA into our DNA, might have played a role in the evolution of life.
A bold idea, challenging the traditional view of genetic information flow.
His work with reverse transcriptase completely flipped the script on how we think about genetics.
It's a reminder that the biological world is full of surprises, and that the boundaries between what we consider living and non-living can be quite blurry.
Well, let's jump into the specifics, starting with Marconi and his amazing journey with wireless telegraphy.
His 1909 Nobel lecture gives us a first-hand account of his early experiments, his struggles, his triumphs, and those unexpected findings that shaped the world of wireless communication we know today.
It's like stepping back in time to witness the birth of a technology that would transform the way we connect with each other.
And to appreciate the scientific landscape of that era.
Right. Back then, radio waves were a relatively new phenomenon, and their behavior was shrouded in mystery.
People were just starting to understand the power of these invisible waves.
Marconi himself describes how he began experimenting on his family's estate in Italy.
Just a young man, tinkering with wires and sparks, trying to transmit signals across his property,
And he quickly discovered that raising the height of his antennas extended the signal's reach.
pushing the boundaries of what was thought possible, sending signals further and further.
He even conducted tests between a ship and a shore station.
A practical application that hinted at the future potential of wireless communication.
And during those tests, he made a fascinating observation.
When the ship was positioned behind a low sand spit, the signal strength would drop noticeably.
It's like the land itself was interfering with the transmission.
This led him to believe that the Earth itself played a role in how radio waves traveled.
Almost like the Earth was acting as a conductor, helping to carry the signal.
That insight is reflected in one of his early patents, where he talks about connecting the transmitter and receiver to the ground.
It's like he was grounding the system using the Earth as part of the circuit.
A brilliant intuition that proved to be essential for long-distance transmission.
But of course, his ideas weren't immediately embraced by everyone.
There was skepticism and resistance from the scientific establishment.
People doubted his claims, especially his assertion that radio waves could travel beyond the horizon.
The prevailing view was that the Earth's curvature would limit long-distance transmission.
Like radio waves would just shoot off into space instead of following the curve of the Earth.
It seemed counterintuitive to think that these waves could bend around the planet.
But Marconi, bless his stubborn soul, wasn't deterred.
He had this unwavering belief in his ideas, this vision of a world connected by wireless communication.
So he set out to do what everyone thought was impossible: transmit a signal across the vast Atlantic Ocean.
A bold and ambitious undertaking, a true test of his theories and his technology.
In 1901, he made history,
From Cornwall, England to Newfoundland, Canada, a signal was successfully transmitted.
spanning thousands of miles, proving that radio waves could indeed conquer those immense distances.
It was a moment that changed the world forever, a triumph of human ingenuity and perseverance,
That single transmission opened the door to instant communication across continents.
laying the foundation for everything from radio broadcasts to the internet we use today.
It's mind-boggling to think that our modern communication technology owes its roots to those early experiments by Marconi.
It's a testament to the power of vision, determination, and the willingness to challenge the status quo.
Now let's shift gears and explore the groundbreaking world of genetic engineering with Daniel Nathans and his work on restriction enzymes.
His 1978 Nobel lecture is a treasure trove of insights into this field, taking us back to the early days of gene manipulation and the revolutionary power of these molecular tools.
What's fascinating is that Nathans wasn't initially setting out to revolutionize genetic engineering.
His primary goal was much more focused, understanding the structure of a specific virus called simian virus 40 or SV40.
AC40, a virus that infects monkeys.
It was a model system for studying how viruses replicate and interact with their hosts.
But along the way, he stumbled upon these remarkable enzymes that would change everything. Restriction enzymes.
Molecular scissors, capable of cutting DNA and specific sequences.
So before restriction enzymes, how did scientists even study DNA? It's like trying to read a book without being able to turn the pages.
They were limited to analyzing whole DNA molecules or using harsh chemical methods to break DNA into random fragments.
Not very precise or efficient.
Right. Restriction enzymes provided the precision tools that scientists needed to dissect and analyze DNA in a controlled way.
Nathans describes in his lecture how he and his colleagues were initially puzzled by the behavior of one particular restriction enzyme called EcoB.
EcoB, an enzyme that required ATP to function, but it didn't seem to cut DNA at a specific site.
ATP, that's the energy currency of cells, right?
Exactly. It was strange that a DNA-cutting enzyme would need this cellular fuel.
And why wasn't it cutting at a specific site? That doesn't seem very useful.
Well, EcoB belongs to a class of restriction enzymes that cut DNA at a random location, some distance away from their recognition site.
So not very predictable or helpful for genetic engineering.
But then they discovered another enzyme, Hindi endonuclease, and this one was a game changer,
Hindi, an enzyme that cut SV40 DNA at a very specific site.
generating predictable fragments, which meant scientists could create a detailed map of the SV40 genome.
It's like having a roadmap to navigate the viral DNA.
And this paved the way for gene cloning, the ability to isolate and replicate specific genes.
Like copying and pasting sections of DNA, a fundamental technique in genetic engineering.
The implications of this technology are mind-boggling.
Gene therapies for treating diseases, genetically modified crops to improve yields, and even the potential to create entirely new organisms.
It's a transformative technology that has raised ethical concerns, but also holds immense promise for addressing some of humanity's greatest challenges.
Now let's shift our focus to the fascinating world of neuroscience and the debate surrounding the neuron doctrine.
It's a story of conflicting views and evolving interpretations within a single scientific discipline.
It highlights the dynamic nature of scientific progress, how our understanding of the world is constantly being refined and sometimes even overturned.
And we'll start with Camillo Golgi's 1906 Nobel lecture, the scientist who developed the silver staining technique that allowed us to visualize nerve cells in detail for the first time.
Golgi's staining technique was revolutionary, allowing scientists to see the intricate structure of neurons, those specialized cells that transmit information throughout the nervous system.
It was a major advance in neuroscience, but ironically, Golgi himself was skeptical of the neuron doctrine.
The neuron doctrine, the idea that the neuron is the fundamental independent unit of the nervous system.
It's the foundation of modern neuroscience, the idea that neurons communicate with each other through specialized junctions called synapses.
But Golgi, based on his observations of stained tissue, believed that nerve cells formed a continuous interconnected network, which he called the diffuse nerve network.
He saw these interconnected structures and interpreted them as evidence against the idea of separate individual neurons.
So on one hand, we have the neuron doctrine championing the idea of discrete neurons communicating through synapses, and on the other hand, we have the reticular theory, supported by Golgi, suggesting a continuous network of interconnected nerve cells.
Two contrasting views of the nervous system, each with its own supporting evidence and passionate advocates.
It's a classic example of how scientific progress often involves clashes of ideas and interpretations.
And sometimes the weight of evidence eventually tips the scales in favor of one view over another.
In the case of the neuron doctrine, further research, using more advanced techniques and experiments, eventually solidified its place as the prevailing view.
But even though the neuron doctrine is widely accepted today, our understanding of the nervous system continues to evolve.
We're still uncovering the complexities of neuronal communication, the intricate interplay between neurons and the brain.
And the role of glial cells, those other cells in the nervous system that were once thought to be mere support structures, but are now known to play a much more active role in brain function.
It's a reminder that even in well-established fields of science, there's always more to learn, always new discoveries to be made.
The journey of scientific exploration is never truly finished.
Now, are you ready to explore the mysterious world of the strong force?
Absolutely. David Gross's 2004 Nobel lecture provides an incredible glimpse into the realm of quantum chromodynamics and the discovery of asymptotic freedom.
It might sound a bit daunting, but trust me, it's a fascinating story. A tale of unexpected behaviors and paradigm shifts in our understanding of the universe.
And it all began with experiments called deep inelastic scattering experiments at the Stanford Linear Accelerator Center, or SLAC.
SLAC, a particle accelerator where scientists smash electrons into protons at incredibly high energies to see what happens.
It's like trying to break open a piñata to see what's inside, but at the subatomic level.
And what they found inside those protons was quite strange.
The way the electrons scattered suggested that the protons were made up of point-like particles.
Tiny, indivisible units of matter.
These point-like particles were eventually identified as quarks, the fundamental building blocks of protons, neutrons, and other particles called hadrons.
Quarks, those elusive particles that come in various flavors like up, down, charm, strange, top and bottom.
It's a whimsical naming convention for some of the most fundamental constituents of matter.
But the way those quarks behaved was a puzzle.
They seem to be bound together incredibly tightly within a proton.
Held together by the strong force, one of the fundamental forces of nature.
But the strength of that force seemed to change depending on how closely the quarks were packed together.
It's like they were free to move around when they were close, but got trapped when they tried to separate.
It was a paradox that challenged our understanding of how forces work.
And that's where asymptotic freedom comes in.
This concept, developed by David Gross and his colleagues Frank Wilczek and H. David Pulitzer, states that the strong force actually gets weaker as the distance between quarks decreases.
It's counterintuitive, right? Usually forces get stronger as things get closer together.
But the strong force is different.
It's like an invisible rubber band that gets tighter as you stretch it, but becomes looser when it's relaxed.
So the closer the quarks are, the less they feel the strong force.
And they behave more like free particles, moving around easily within a nucleus.
This explains why those early experiments at SLAC found that quarks seem to act like free particles when probed at high energies.
They were essentially seeing the quarks in their relaxed state when the strong force was weak.
But as soon as those quarks try to separate, the strong force kicks in and pulls them back together.
It's like they're confined within the nucleus, unable to escape.
This is known as quark confinement, a phenomenon that is still not fully understood.
It's a major mystery in physics, a puzzle that continues to intrigue scientists.
We know that quarks are always bound within hadrons, like protons and neutrons, but we don't yet have a complete theoretical explanation for why they can't exist in isolation.
It's like a cosmic rule that prevents these fundamental particles from roaming freely in the universe.
But the discovery of asymptotic freedom was a major breakthrough.
It revolutionized our understanding of the strong force and its role in shaping the universe.
From the formation of atomic nuclei to the behavior of matter under extreme conditions, asymptotic freedom has helped us unlock many secrets of the cosmos.
Okay, before we dive into the fascinating world of retroviruses and the origin of life, let's take a quick break.
We'll be back soon to continue our exploration of these amazing Nobel lectures.
Stay tuned for more mind-bending scientific adventures.
Welcome back, everyone. Ready for more Nobel Prize-winning science?
You bet. Where are we headed next on this incredible journey?
Let's dive into the world of retroviruses and the protovirus hypothesis, a fascinating theory about the origin of these unique viruses.
Okay, so before we get into the specifics of Howard Timmons' work, maybe we should set the stage a bit.
Good idea. Back in the early 1970s, the central dogma of molecular biology was pretty much the law.
Oh, right, the central dogma.
DNA to RNA to protein, that was the flow of genetic information, no exceptions.
DNA is like the blueprint, RNA is the messenger, and proteins are the workers, right?
Exactly. But then retroviruses came along and kind of threw a wrench into that neat picture.
Retroviruses, they're the ones with RNA as their genetic material instead of DNA, right?
That's right. And what's even weirder is that when a retrovirus infects a cell,
It does something pretty unexpected, right?
yeah, it converts its RNA into DNA, which then gets integrated into the host cell's genome.
Hold on, it's like going backwards. It's like rewinding the tape of genetic information.
Exactly. It completely challenges the central dogma, and that's why Timmons' work caused such a stir in the scientific community.
So how did he figure this all out? Was it like a sudden eureka moment, or was it a more gradual process?
Well, Timon, along with another scientist named David Baltimore, they both independently discovered an enzyme called reverse transcriptase.
Reverse transcriptase.
Yeah, and this enzyme is found in retroviruses, and it's what allows them to do this crazy backward conversion of RNA to DNA.
Wow, so this enzyme basically flipped the script on how we thought genetic information worked.
Absolutely, it was a paradigm shift in our understanding of genetics.
Okay, so we've got these strange retroviruses that can turn their RNA into DNA.
But where did they come from in the first place? That's the question Timon was trying to answer.
And that's where the protovirus hypothesis comes in, right?
You got it. Timon proposed that retroviruses, way back in evolutionary history, they originated from normal cellular RNA molecules.
Normal RNA molecules, like the ones our cells use every day.
Exactly. But somehow, these RNA molecules acquired the ability to replicate independently.
So these rogue RNAs basically went rogue and became viruses.
It's like a cellular rebellion. And over time, they evolved this amazing ability to convert their RNA into DNA, sneak into the host cell's genome, and eventually become the retroviruses we know today.
It's like a cellular espionage story.
That's a great way to put it. And to support his hypothesis, Timon pointed to evidence from a virus called spleen necrosis virus, or SNV.
SNV. What's special about that virus?
Well, Timon and his colleagues found that SNV virions, those infectious particles released by the virus,
Right, virions, those tiny packages of viral mischief.
they contained RNA polymerase activity.
Wait, RNA polymerase, isn't that the enzyme that normally transcribes DNA into RNA?
That's the usual role, yeah.
Mhm.
But in SNV virions, this RNA polymerase was doing something different.
It was like the enzyme was moonlighting, taking on a new job.
Exactly. It was involved in synthesizing a small RNA molecule, not a full-length RNA copy of the viral genome.
Okay, so how does this tie into the protovirus hypothesis?
Timon argued that this RNA polymerase activity in SNV variants could be like an evolutionary leftover from the original protovirus.
A kind of molecular fossil, a hint of their past life as normal cellular RNA.
Exactly. A trace of their ancient origins.
So the idea is that these protoviruses, way back when, they had this RNA polymerase activity as part of their normal cellular function.
But as they evolved, they figured out how to use reverse transcriptase to create DNA copies of themselves.
It's like they upgraded their toolkit, adding this new weapon to their arsenal.
Precisely. And this DNA then got integrated into the host cell's genome, eventually leading to the retroviruses we see today.
So Timmons' work on retroviruses has had a huge impact, not just on how we understand virus evolution.
But also on the evolution of genomes themselves. It's like a whole new layer of complexity in the story of life.
It makes you wonder how many other surprises are hiding in the genomes of different organisms.
Absolutely. And it reminds us that the boundaries between viruses and cellular life can be quite fuzzy.
It's like this constant interplay between different forms of genetic information.
A constant dance of evolution and adaptation.
Okay, I think my brain needs a break from viruses for a moment.
Me too.
Hmm.
How about we shift gears and explore the world of atoms?
Sounds good to me. Atoms, the building blocks of everything we see and touch, they're always fascinating to explore.
We're in for a treat with Niels Bohr's 1922 Nobel lecture. It's a journey back to the early days of atomic theory and the development of the quantum model of the atom.
The quantum model, that's the one with electrons orbiting the nucleus in specific energy levels, right?
That's the one. But back in the early 1900s, that picture was revolutionary.
Before Bohr came along, what did people think atoms looked like?
The prevailing model was called the plum pudding model.
The plum pudding model, that sounds delicious.
It does, but it wasn't very accurate. It envisioned the atom as a sphere of positive charge with negatively charged electrons embedded within it, like plums in a pudding.
So a kind of diffused blob of positive charge with electrons scattered throughout.
Right. But this model couldn't explain certain things that scientists were observing,
Like what?
like the line spectra emitted by atoms when they're heated.
Oh, right, the line spectra, those unique sets of colors that each element emits, like fingerprints for atoms.
Exactly. But if electrons were just randomly distributed within a blob of positive charge, why would atoms emit specific colors?
It's like the plum pudding model couldn't account for those atomic fingerprints.
And that's where Bohr's model came in.
So Bohr's model, it had this radical idea about electrons orbiting the nucleus.
Right. But not just orbiting randomly. Bohr proposed that electrons could only exist in specific quantized energy levels.
Quantized.
Yeah, it means that the energy of an electron isn't continuous. It's like a staircase. Electrons can only be on specific steps. They can't be in between.
So it's like those electrons are restricted to certain energy levels, like rungs on a ladder.
Exactly. And this idea of quantized energy levels, it was revolutionary.
And how did it explain the line spectra?
Well, Bohr proposed that when an atom is heated, its electrons can jump to higher energy levels by absorbing energy.
Like climbing up the rungs of that energy ladder.
Right. And then when those electrons fall back down to lower energy levels, they release energy in the form of light.
And the color of that light depends on the energy difference between the two levels.
So each line in an element spectrum corresponds to a specific electron transition.
It's like each color is a signature of a particular electron jumping between energy levels.
Precisely. And that's why each element has its unique line spectrum, because its electrons are arranged in unique energy levels.
It's amazing how something as seemingly simple as the color of light can reveal so much about the structure of atoms.
It's a testament to the power of spectroscopy, the study of how matter interacts with light.
Okay, so Bohr's model explained the line spectra, but didn't it also solve another problem?
You're thinking about the problem of why electrons don't just spiral into the nucleus.
Right. According to classical physics, an electron orbiting a nucleus should lose energy and eventually crash into the nucleus.
It's like a satellite falling out of orbit.
But atoms are stable, so something else must be going on.
And Bohr had an answer for that too.
So what was his explanation? Why don't electrons just spiral into oblivion?
He proposed that electrons in their lowest energy state, called the ground state, they don't radiate energy.
So they're in a kind of stable equilibrium, not losing energy as they orbit.
Exactly. They're in a kind of quantum safe zone.
And electrons in higher energy levels, they can only jump down to lower levels, releasing energy in the process.
They can't just gradually lose energy and spiral inward.
It's like those electrons are stuck on those energy rungs. They can't just slide down the ladder.
So Bohr's model, even though it was still a simplified picture of the atom, it explained a lot of things that the plum pudding model couldn't.
It was a major step forward in our understanding of the atom, and it laid the groundwork for the development of quantum mechanics.
The theory that revolutionized physics in the 20th century.
But even revolutionary theories have their limitations, right? Bohr's model wasn't perfect.
True. It couldn't fully explain the spectra of atoms with more than one electron, and it didn't account for the wave-particle duality of electrons.
The wave-particle duality, that's the idea that electrons can behave like both waves and particles, right?
Exactly, a concept that is fundamental to quantum mechanics.
So Bohr's model was a stepping stone towards a more complete and accurate picture of the atom.
It was a crucial step in our journey to understand the fundamental building blocks of the universe.
Well, Bohr's model may have had its limitations, but it certainly sparked a revolution in physics.
And it's amazing to think that so much of our modern understanding of chemistry, material science, and even technology stems from those early insights into the structure of the atom.
Now, before we move on to our next scientific adventure, I'm curious.
What's on your mind?
Bohr also talks about X-ray spectra in his lecture, right? How did those fit into the picture?
X-ray spectra. They played a crucial role in confirming Bohr's model of the atom.
X-rays, they're a form of electromagnetic radiation, right?
Yeah, like light, but with much shorter wavelengths.
So they can penetrate matter and interact with the inner parts of atoms?
Exactly. By analyzing the X-ray spectra emitted by different elements, scientists could figure out how the electrons were arranged within those atoms.
So X-ray spectra were like another type of atomic fingerprint.
Precisely. And what those fingerprints revealed was that the energy levels of electrons in an atom are indeed quantized, just as Bohr had predicted.
It was like having independent confirmation of his theory.
And it helped to solidify the idea that electrons can only exist in specific energy states, not just anywhere within the atom.
So X-ray spectra were like a window into the quantum world, revealing the discrete nature of electron energy levels.
A powerful tool for probing the inner workings of atoms.
It's amazing how these different techniques, from studying visible light to X-rays, all converge to support Bohr's model.
It's a beautiful example of how science often progresses with different lines of evidence coming together to paint a more complete picture of the universe.
Well, we've covered an incredible amount of ground in this part of our deep dive, from the unexpected behavior of retroviruses to the revolutionary model of the atom.
It's been a whirlwind tour of scientific breakthroughs.
We'll continue our exploration in the final part of this episode.
Get ready for more mind-blowing insights into the world of science.
All right, we're back and ready to wrap up our epic deep dive into these Nobel lectures.
I'm excited to see what scientific wonders we uncover in this last batch.
Me too. So where are we headed first?
Let's start with something pretty fundamental.
Yeah.
How cells talk to each other.
Cellular communication. I know cells send signals, but what's the actual mechanism? It all seems kind of mysterious.
Well, that's where Earl Wilbur Sutherland Jr. comes in. He won the Nobel Prize in Physiology or Medicine in 1971 for his work on how hormones work.
Hormones, those chemical messengers that travel throughout the body, regulating all sorts of processes.
Exactly. Sutherland's research uncovered a key molecule called cyclic AMP, or CAN-AMP, that plays a crucial role in how cells respond to hormones.
CAN-AMP, so it's like a middleman, relaying messages from the hormone to the inner workings of the cell.
That's a great way to put it. Before his work, scientists knew hormones could trigger changes in cells, but the how was a bit of a black box.
It's like knowing someone knocked on the door, but not knowing how that knock led to actions inside the house.
Precisely. Sutherland's research showed that when a hormone binds to its receptor on the cell surface, it triggers the production of CAM-AMP inside the cell.
So the hormone is like the first messenger arriving at the cell's doorstep.
And then CAM-AMP acts as the second messenger, relaying that message to the cell's interior and setting off a cascade of events.
So it's a two-step process, a molecular relay race.
Exactly. And his discovery of CAM-AMP as a second messenger was huge for cell biology.
It's like he cracked the code of cellular communication.
It opened up a whole new understanding of how cells respond to their environment and regulate all sorts of processes.
From metabolism to memory formation, it's all connected.
And it's not just CAM-AMP. Sutherland's work paved the way for discovering tons of other signaling molecules and pathways.
Revealing this intricate web of communication within our bodies.
It's like a complex symphony of molecular signals orchestrating all the functions of life.
Okay, let's shift gears from the microscopic world of cells to something completely different.
Where to?
How about something mind-boggling? Superfluidity.
Ah, superfluidity. A truly bizarre state of matter. I think you'll enjoy this one.
I have to admit, I don't know much about it. What makes it so special?
Well, we'll be exploring this phenomenon through the work of Piotr Kapitsa, who won the Nobel Prize in Physics in 1978 for his discoveries in low-temperature physics.
Low-temperature physics, so we're talking about some seriously cold stuff.
Extremely cold.
Yeah.
We're talking about liquid helium cooled to just a few degrees above absolute zero.
Wow, that's mind-numbingly cold.
And at those temperatures, something incredible happens. Liquid helium becomes a superfluid.
A superfluid?
Yeah, it means it flows without any resistance.
No resistance at all, but everything experiences friction, right? Like, if I slide a book across a table, it eventually stops.
That's true for normal fluids, but superfluids are different. They can flow through tiny capillaries without losing any energy, defy gravity by climbing up walls.
Wait, defy gravity?
Yeah, it's like they're immune to the usual laws of physics.
It's like something out of a science fiction movie?
It is.
Yeah.
And it's all thanks to the weirdness of quantum mechanics.
Quantum mechanics, that branch of physics that governs the behavior of matter at the atomic and subatomic levels.
Right. At those super low temperatures, the atoms in the superfluid start behaving more like waves than particles.
So it's like their wave-like nature takes over.
And they all start moving in sync, forming a sort of collective entity, a macroscopic quantum state.
So instead of individual atoms bouncing around randomly, they're all moving together in a coordinated way.
Exactly. And that coordinated movement, governed by quantum rules, is what gives superfluids their bizarre properties.
It's like a synchronized dance of atoms gliding effortlessly through space.
A beautiful and mysterious phenomenon that continues to fascinate physicists today.
Okay, my brain is officially boggled, but before we call it a day, let's take one last journey.
Sounds good to me. Where are we headed?
How about to the beginning of everything, the Big Bang?
The Big Bang. the ultimate origin story, a perfect way to end our deep dive.
We'll be exploring this momentous event through the work of Arno Penzias and Robert Wilson, who shared the Nobel Prize in Physics in 1978.
They discovered the cosmic microwave background radiation, often called the CMB, which was a game changer for cosmology.
So the Big Bang Theory, it's the idea that the universe started as an incredibly hot, dense state and has been expanding and cooling ever since, right?
Exactly. But before Penzias and Wilson's discovery, the Big Bang was just one of several theories about the universe's origin.
So what made their discovery so special? What is this cosmic microwave background radiation?
Well, it's a faint glow of microwave radiation that permeates all of space.
Microwave radiation, like what we use to heat up food.
The same kind of radiation, but much, much weaker. And it's coming from every direction in the universe.
It's like a cosmic background hum.
And according to the Big Bang Theory, this radiation is the afterglow of the Big Bang itself, a remnant of that primordial fireball.
So it's like we're seeing the faint echo of the universe's birth.
Exactly. As the universe expanded, this radiation stretched and cooled, shifting to longer wavelengths and eventually reaching the microwave range we observe today.
So it's a fossil record of the early universe, a snapshot of what things were like billions of years ago.
And its discovery provided the strongest evidence yet for the Big Bang Theory.
But how did they even find this radiation? It sounds incredibly faint.
It was a bit of an accident, actually. They were using this sensitive radio antenna at Bell Labs.
Bell Labs, the birthplace of so many technological innovations.
Right. And they kept picking up this persistent hiss in their data, no matter where they pointed the antenna.
It was like unwanted noise.
They tried everything to get rid of it, even cleaning pigeon droppings off the antenna.
Pigeon droppings. That's pretty down to earth compared to the cosmic origins of the universe.
It is. But after ruling out all other possibilities, they realized that this hiss was actually the cosmic microwave background radiation.
Wow, talk about finding a needle in a haystack.
It was an incredible discovery, a serendipitous moment that changed our understanding of the universe.
It confirmed the Big Bang Theory and opened up new avenues of research in cosmology.
We've come a long way since then, mapping the CMB in detail and using it to learn about the age, composition, and evolution of the universe.
It's amazing how much we can learn about the universe from this faint echo of its beginnings.
It's a testament to the power of science and the human drive to explore the unknown.
Well, I think we've reached the end of our extraordinary journey through a century of Nobel Prize-winning science.
From the tiniest molecules to the vast expanse of the cosmos, we've covered a lot of ground.
We've witnessed the power of human curiosity, the thrill of discovery, and the enduring quest to understand the universe and our place within it.
It's been an incredible privilege to share this journey with you all.
And we hope it's inspired you to keep exploring, to keep asking questions, and to never stop marveling at the wonders of science.
Because the world of science is vast and full of mysteries, and there's always something new to discover.
So until next time, keep that scientific spirit alive and well.